Good afternoon to those of you on the East Coast. Good morning to those of you on the West Coast, and good day to those of you in the middle. I'm John Anderson, President of the National Academy of Engineering, and I thank you for joining the second pitch showcase of the NAE's call to action for engineering solutions to the COVID-19 pandemic. The NAE launched the call for engineering action in April to promote the brainstorming of ideas that could protect public health and the economy during the pandemic. More than 800 individuals have responded to the call, including NAE Grand Challenges Scholars Program students and alumni, mid-career professionals associated with our Frontiers of Engineering program, and members of the National Academy of Engineering. I want to especially thank the COVID-19 Call to Action Executive Committee, members of the Expert Review Committee, and all the GCSP student volunteers who helped run the initiative. Special thanks to the LA node of I-Corps and to the donors who have contributed resources to make this happen. Today we will showcase 5 concepts that have gone through the process and hopefully launch their ideas. You can learn more about the call to action and participate in it by visiting our website, which is supposed to be shown on the screen. Now I will turn things over to Dr. Alice Liu, who will moderate today's event. Alice is Assistant Director for the Office of Technology, Innovation, and Entrepreneurship at the University of Southern California's Viterbi School of Engineering. Over to you, Alice.
Thank you, John. This is an exciting and important initiative. I'm so pleased to moderate this pitch showcase again and showcase new technologies being developed to combat the COVID-19 pandemic. If you joined us for the first event, you already know the drill. As a reminder, the format for today, each team will be giving a 5-minute pitch, followed by 8 minutes of Q&A with our Expert Review Committee. We refer to them as the ERC. The ERC are not judges, as this isn't a competition. Our goal here today is to help advance these ideas and accelerate their trajectory to reality. This includes your participation, our audience viewing from home. If you have comments or thoughts, or ways to help as you're listening, we encourage you to tweet about this event and use hashtag NAECOVID19. You can also send your comments to engcovid19@nae.edu. So for today's ERC, we are honored to have a very distinguished panel of reviewers. In total, we have 10 reviewers who have been divided across the Q&A sessions. I will introduce our ERC panelists at the start of Q&A. Okay, that sets our ground rules. I think we're ready to begin. I now would like to welcome up our first team. Our first team is Self-disinfecting Masks with Nissim presenting. Nissim, you have 5 minutes.
All right. Hello, everyone, and good afternoon. This is Nissim from Georgia Tech, and I'm presenting on behalf of VoltaPure. I'm going to be talking about our self-cleaning reusable mask technology, which is going to be an important step in addressing this COVID crisis. But first, let's look at the problem. Next slide, please. So as you know, there has been a long-term mask shortage over the last few months. These are headlines from less than 1 month ago. And projections say that this is going to be a long-standing issue throughout the crisis, running into a demand of millions of masks per week. So one way to solve this crisis is to reuse masks. Unfortunately, existing approaches to doing this are either inappropriate, inconvenient, or inaccessible. For example, the ventilation approach, which you see everyone using by just hanging their masks all over the place, is actually often counterproductive because it can release the pathogens back into the air. Solar disinfection tends to do a slightly better job of actually killing the pathogens, but that is often hard to do if there's no sunlight, or is very time consuming even if there is. Ultraviolet light and chemical disinfection tend to be a lot more effective, but naturally they are out of the reach of most consumers. And actually in cases like chemical disinfection, it can end up actually harming the fabric of the mask, or even leaving disinfection residues that can be harmful to the user. Next slide. So our solution to this is self-disinfecting masks, which are based on our own technology. The beauty of this solution is that it is a physical form of disinfection, and hence there's no chemical byproducts to worry about. This involves using a low voltage across the mask, and hence has low energy consumption and can be rigged to a battery or even like a dynamo, human powered. This is reusable because it kills viruses and bacteria, and this is a novel technology whose patent we have applied for. As you can see in this microscope image, this technology involves our locally enhanced electric field treatment, which consists of copper nanowires that are threaded in with the fabric of the mask. And what these nanowires do is to concentrate the electric field at their tips, which means that any pathogens that come in contact with them are neutralized by electrophoresis. Next slide. This is the solution with which we went into the program, and the key developments here that we've made over the summer are to discover 2 customer segments as well as a potential beachhead market. As for the customer segments, one important segment is essential workers, which represent high frequency usage, 3 to 4 times, around 6 to 8 hours a day for 7 days a week. But this is also a more price sensitive segment, since these purchase decisions are made in bulk by the higher ups within the organization. The general public is the other segment, which is lower frequency usage, around 3 to 4 times a week and a few hours at a time. And naturally, they are a little more open to spend because they are one-off purchases being made by the users. Now, the potential beachhead market, which we discovered by conducting more than 15 direct interviews with potential customers, is elderly care facilities. Now, there are over 15,000 elderly care facilities in the US, according to the CDC, which tend to over 1 million patients, which represents a large market, again, running into a demand of millions of masks. And we had the opportunity to interview the vice president of one such facility, which has over 100 locations all over the southern United States, and is home to 8,000 plus residents and has had 300 plus cases over the last few months. What we learned by talking to them was that not only do these facilities have a susceptible population and PPE shortages, but they do tend to have stricter reimbursement policies compared to most hospitals. However, this is a big problem for them, and they are open to trying new products, including a product like ours, and that might give us a good entryway into the market. Next slide. What we intend to do over the coming months is to prototype our technology and get like a working prototype, which we can eventually test in the lab to understand its exact disinfection capabilities under different conditions, and then pilot test it with the initial user base. We are on the business front urgently looking for a business mentor to help us scale this technology. We are going to be applying for programs such as the NSF I-Corps, through which we want to develop a business model. And eventually we will partner with manufacturers who, based on our IP, will manufacture the product so that we can get it to the market. Next slide. This is the team that has developed this product. As you can see, between Dr. Shea, Jianfeng, and myself, we represent a pretty diverse group of backgrounds, and we come at this problem from different angles. But we are very much in need of a business mentor, preferably someone with biomedical experience, healthcare connections, and manufacturing know-how. So if you can help, or if you know someone who can help, we would love to hear from you about that. So that said, I would like to open the floor for questions. Thank you for listening.
At this time, I'd like to introduce our, thank you. At this time, I'd like to introduce our ERC panel for this team. I'll be giving a short bio, but fuller bios can be found on the NAE website. We have, for this Q&A session, Neil Payton, former Vice President of Technology at Helmet Corporation. We have Paul Citron, former Vice President of Technology Policy and Academic Relations at Medtronic. We have John Martin, former Chairman and CEO of Gilead Sciences. We have Darlene Solomon, Chief Technology Officer and Senior Vice President of Agilent Technologies, and Larry Seifert, former Vice President at AT&T. Reviewers, you have 8 minutes for Q&A. And the first question goes to Larry.
The structure, is this something that could be adapted to existing masks, or is it technology, or is it something that we have to create from anew, if you will? And that would affect, obviously, your distribution plans and the like. sounds fascinating technology. The question really is from the implementation standpoint.
Right. So yeah, that's a great question. Thank you. In fact, the electrodes that we have been working with so far have largely been like flexible sheet-based electrodes, and hence we have reason to believe that we might be able to, as I mentioned in one of the earlier slides, be able to kind of alternate them with the fabric of masks. Now, we are still looking into what exactly the mask manufacturing process looks like to understand how easy or difficult it would be to add a step like this to the manufacturing process. But we have reason to believe that it can be retrofitted onto existing mask manufacturing.
Thank you. Our next question comes from John.
Excuse me, Nissim. That was a very well organized talk for only 5 minutes. Congratulations.
Thank you.
And what you're doing makes a lot of sense if you have the price in the range, as you just discussed, to be able to compete with multiple discardable masks. That gives you some flexibility. I'm curious, being in Atlanta, and with all the issues we see globally about certification of masks and proper use of masks and imperfectly made masks, if you've considered working with the CDC to get some early guidance and integration of them into what your plans are.
Right. Yeah, thank you. That too is a good question. We haven't yet started working with any of the regulatory bodies, but we do believe we will have to deal with them sooner or later. And hence, in the request for a business mentor, preferably we're looking for someone who can help us navigate all of that. But well, this is a technology that, as you can see, was something we developed for water treatment originally. And theoretically, this is something that should be able to translate to aerosol disinfection. So as we are kind of setting up our working prototypes, we want to understand first what levels of disinfection we can achieve under what conditions before going to the regulatory authorities. I hope that answered your question.
Perfect. Thank you.
Thank you. Our next question goes to Paul.
First of all, again, congratulations. It's I had to learn something about the technology because the technology was new for me, but it looks very promising and exciting. I actually have a number of questions, but to answer one of your questions, have you approached the business school at Georgia Tech? They have an entrepreneurship program that might be able to give you some mentorship right on campus.
So yes, in fact, we have begun speaking to them. They too are on the lookout for a mentor for us right now, but they didn't have anyone who kind of met the conditions that we, or rather just the pointers that we had given them. So we're yet to hear back from them. Maybe Jianfeng, you can talk a little more about this since you've been interacting with them more regularly.
Yeah, sure. This is Jianfeng from Georgia Tech. So we have already got connected with Venture Lab in Georgia Tech. So we have several connections there. And also I know there's a program with the business school called like Tiger program. So they can connect one like business school students with us, with our engineers and scientists to like do this entrepreneurship training, something like that. Yeah.
And I have another question, if I might, Alice, and that is,
Please.
A lot of the masks are layered technologies, and a lot of the masks that the public uses are do-it-yourself masks where people sew them at home. So can your technology be reduced to a sheet of nano wires that could be on some kind of a substrate that would be imported or integrated into a do-it-yourself multi-layer mask?
Yeah, that's actually a very interesting spin-off idea or like new angle for looking at this. We haven't considered this. The obvious challenges that come to mind while answering that would be that it's an electrical solution, so it would also involve having kind of a battery connection. So it would work better if it were integrated with the framing of the mask and all of that. But I do think it might be possible to maybe ship it such that we just send them the electrode, which is flexible, maybe sandwiched between just two cloth sheets, and then they can retrofit that onto their homemade masks.
Great. Our last question will go to Darlene.
Great, thank you. And again, echoing really nice presentation, and most importantly, it really seems like this is a technology where maybe this is going to say the right technology at the right time. And along those lines, I'm interested in electrical disruption of pathogens has been around for some time, and can you speak to maybe why it hasn't been adopted significantly up until this point, and some of the ways in which this technology really enables that to happen?
Right. Sure. I'll speak about a few of the technicalities, and then I think I'll let Dr. Shea also elaborate a little bit on this, since he is our PI and research head. But well, a short answer is that in our technology, the advantage here is that since we have nanowires, what we rely on is even a small electric field, which can be delivered by just your portable batteries, can be concentrated by those nanowires to something strong enough to actually electroporate the bacteria. So one possibility is that since it usually takes larger currents to kill bacteria effectively at scale, maybe that's why this hadn't been shrunk down so far. But yeah, Dr. Shea, do you have anything to say about this?
Yeah, sure. So let me just a little bit. So basically, I agree with the question that basically using electric field to kill pathogen has been actually applied in some field. For example, in the food pasteurization, they use electric field to treat the liquid food. But the problem is that usually they have to use a very high voltage to achieve the enough electric field strength to kill the pathogens. The unique technique in our case is that we use nanowires to modify the electrode surface. So that's why we only need to apply a very small voltage, just like a few volts, and then the electric field concentrated near the tips of these nanowires can be strong enough to kill the pathogen. So actually, when we do the treatment, we are not creating a very uniform, very high electric field anywhere, but it's only on the electrode surface near the tips. So when we use this technology in a mask or in the filter, we not only collect those pathogens, but we also kill these pathogens on the electrode surface.
Great.
Very helpful. Thank you. With the lower fields, though, I presume there is some additional capability and demonstration of, the efficacy and so forth that, will be part of the next steps.
Yeah, that's what we're doing. Yeah, thank you.
Good. Thank you.
Great. Thank you, reviewers. Thank you. We also hope today's conversation will spark new ideas and initiatives. If you've been inspired, please consider answering the NAE call to action at nae.edu/COVID-19 and enter with your own idea. The incubator is still going on, and we will be doing more of these pitch showcases. I'd now like to introduce our next team, Wearable Patch for Remote Monitoring of COVID-19 Patients. Emily, are you ready?
Hi, everybody. Our team, Postlytics, is very excited and honored to present to you all today. We will begin with an introduction to our team. Next slide, please. We are all students who attended the Global Grand Challenges Summit last September. Postlytics was one of the winning finalists of the Innovation Hackathon organized by the RAE. We span 3 different countries, 5 different time zones, and we have a range of technical and business focuses on our team. Next slide. We will begin with an introduction to the problem. Pre-COVID-19, overcrowding of hospitals was already an issue. If we take a look at the stat on the top, we can see that there are only 2.8 beds per 1,000 people in the US. In and of itself, hospital overcrowding leads to inefficient and decreased patient care. This hospital overcrowding is exacerbated by COVID-19. As shown in the graph, COVID-19 has led to a sharp increase in hospitalization rates. And one example from last month in Houston is a lack of ICU beds that led to no choice but to board patients in the ER. Next slide, please. So now to dive deeper into the problem, if we take a look at the right, pre-COVID-19, 90% of emergency departments reported crowded conditions. On the left, some causes of this point to fewer emergency room beds, growing patient volumes, and patients coming in with avoidable visits. These causes indicate that a broader hospital-wide solution is needed. This is a concern on the hospital's end, but also to the patient's end, especially in this pandemic. Individuals with pre-existing health conditions are skeptical of returning to hospitals for regularly needed treatment. Severe heart attacks being treated in the US are down by nearly 40%. Healthcare professionals are worried patients are dying in their homes and have no idea about their condition. A solution to hospital overcrowding and greater information flow from a remote location is needed. Let's get into our solution. Next slide. On a high level, we aim to increase remote monitoring capabilities so the patient's recovery location is now at home versus in the hospital, and the patient will still receive the support of being monitored by healthcare professionals in real time, where instead of decentralized patient data can be accessed more easily and used to inform future care. Next slide, please. Our specific solution innovates in 3 ways. 1, we have the novel wearable design using leading edge materials for enhanced reusability and flexibility. 2, unique techniques for real-time data transmission. And 3, utilizing analytics and machine learning algorithms to inform both healthcare professionals and patients themselves. Next slide, please. There are multiple potential customers Postlytics can impact, the focus being on hospitals and clinics in response to the current pandemic and our interview feedback so far. Other paths to market may include device direct-to-patient to sales of big data. Let's now talk about some of the benefits. Next slide. So there are a wide range of benefits as well as direct cases for COVID-19 for each of these potential customers. To highlight a few, Postlytics can help increase bed space through potential early discharge, reduce readmission rates by ensuring patient wellness post-discharge, and increase worker safety for industry military employees. Next slide, please. As a status update, since the GGCS, we've invested in diversifying the team's expertise, received seed funding from the RAE, won the Ingenuity Lab competition at the University of Nottingham. As part of this initiative, completed the I-Core training, and we've now begun prototype fabrication. Next slide. Postlytics is continuing to move forward. On the top slide, we see that the technical timeline includes building our prototype and MVP, applying for applicable patents and the National I-Core program, and beginning the FDA approval process. On the business end, we plan on continuing to conduct customer interviews and market research in order to finalize our business model and then move into launching our pilot program. Next slide. And finally, the asks that we have at this point are potential contacts for customer interviews and partnerships. None of us have direct experience in the FDA or patent process, so we are seeking mentorship, pro bono legal support, or funding. With that, thank you all very much for your time. We are now open for questions.
Thank you, Emily. I'd like to welcome up our ERC panel for this team. We have Roderick Pettigrew, CEO of nHealth, a multi-college engineering health initiative between Health Science Center and College of Engineering at Texas A&M. Dennis Disher, Professor of Chemical and Biomolecular Engineering at UPenn. Sam Petrotta, Chairman of the Petrotta Group. Monty Alger, Professor of Chemical Engineering at Penn State University. Kurt Peterson, Co-Chair of the Hard Tech Group in the Silicon Valley Band of Angels. It's Silicon Valley's oldest angel investment group. Again, if you have questions, reviewers, please use the raise hand function. So we are ready to begin, and the first question will go to Roderick.
Thank you, Alice. First thing I want to do is congratulate the team. I noticed that the team is assembled from students that come from 3 different countries, multiple institutions, and that you met at the last NAE Grand Challenges Summit. So that is exactly what we had hoped would happen at this type of event, and that it would in fact stimulate this kind of collaborative activity. So congratulations on that front. Also, you do address a significant problem that probably might fly under the radar a bit. So hats off to you in identifying a unique area where you can help. Two questions. What are the vital signs that you actually will be able to monitor? You showed a strip that indicated that you were looking at some aspect of the ECG or a heart rate, but I'd like to know specifically what vitals you are monitoring on the front end. Then on the back end, tell us a little bit more about the software. You mentioned using artificial intelligence to do your analytics, but are you developing the software as well? And what specifically would that provide to the end user?
Jessica, do you want to start?
Yes, thank you, Emily. And thank you so much for those questions. I will be speaking in terms of the vitals that we are currently hoping to get working. One of them being the ECG sensors, so we are currently in development of those sensors. Another is the body temperature. We also have a tilt temperature in order to track the body position of the patient. And in addition to that, we are looking at position as well. Yes.
And the software, the analytics?
Luke, would you like to cover the analytics portion?
Yeah, are you guys developing the analytical package that you present to whoever would purchase this?
Yes, absolutely. I will be taking the software part. So basically, our project will include two parts of analytics. The first is descriptive analytics, and second is preventive analytics. So for the descriptive analytics, we decided to adopt a data visualization software that already exists in the marketplace, such as Tableau, such as Power BI, MicroStrategy, along with our customized database architect and database design. And for the later part, the preventive analytics, I think we will also adopt some open source or personally developed some machine learning algorithm to predict the future occasions based on the existing and probably the real-time data set that I extract from the vital patch. Thank you.
Thank you. Our next question comes from Kurt.
Yeah, congratulations again on actually, this is a great application, I think, for this COVID crisis, and congratulations on coming up with a great idea. So my question revolves around IP that's already in this area. There are quite a number of companies that are making medical patches, including ECG, temperature, SpO2, inertial devices for position. So my question is around, so what kind of unique IP do you folks have for this type of patch?
So I can answer that. So what makes Processix unique is one of the things, the recyclability aspect of it. So we're tending to design it so it could be fully disassembled easily, as well as all the parts can be replaced. We're using technology we're using is liquid metal for the circuitry, PDMS polymer, which I believe is fully recyclable, and so whereas on other biosensors that we've researched currently, such as Philips and Vital Patch, I believe, which currently are our competitors, we believe do not offer that. Also, the live data transmission, which we offer with the use of our data analytics, and yeah, we're hoping that will make us stand out from the market of our competitors.
To add on to that, so some of the main aspects is, one, the reusability aspect is something that we're working hard on to help make the remote monitoring more sustainable and able to be used on a more of a service schedule or larger scale. Other aspects of that also include the materials that we're using for the patch itself are going to be specifically designed for greater flexibility. So that's something that's unique from things on the market currently. But we also see applications for patent in the manufacturing process. The way that we're putting the materials together may have some unique IP related to it, and also on the software end with the algorithm having some unique IP there as well.
Thank you, Emily. Our next and last question will come from Dennis.
Yeah, thanks. Congratulate the team also. It's an interesting complementary proposal to COVID, but let me come back to the sensors, the detection that temperature, blood pressure, heart rate that you list in your abstract, but maybe possibly more, and ask whether any of those are especially pertinent to some of the patient monitoring you'll do in the case of patients actually having COVID-19. Certainly higher temperature is something, but you're not really seeming, I guess, to target COVID-19 disease patients. But if you could, right, would you prioritize some of these measurements over others and, analytically detect it? Have you given thought to that?
Yes, thank you so much for that question. Right now, we are in the developing stages of trying to produce a product that is very general. The selection of those 5 sensors were due to the fact that we wanted to design something that was going to tackle multiple illnesses. So that is definitely something moving forward we will take into consideration, is designing a prototype that is specifically will be used for COVID and monitoring COVID itself.
Yes, and to add on that, our innovation is not only targeted at people who have already infected coronavirus, but also someone who has a high risk by going to a hospital. So our product can prevent them from going to a hospital instead of having the higher risk of infection. So they can have the kind of the detection at home, so that it can avoid going to overcrowding hospital and avoid problem like that.
Yeah, I understood that to be the emphasis rather than more COVID-19 targeted patients, but it sounds like you're thinking of both. So thanks a lot.
Great, thank you team, and thank you reviewers. Those of you in the audience, if you have ideas for this team or have ways to help, or you have any comments for the NAE, please contact the NAE at engcovid19@nae.edu, or you can tweet #NAECovid19. I'd like to introduce our third team for today. Our third team is early COVID-19 detection and neurovascular recovery in pre-symptomatic and asymptomatic individuals.
Thank you, Alice. What I'd like to share is I'm at a hotel in Palo Alto, California, where my family lives, after driving 1,800 miles from San Antonio, Texas, where my lab members and I live. In the process, I had to be very careful about safety and making sure I was not at risk for exposing myself to COVID-19, because I was traveling as fast as possible because of a medical emergency with my nephew. So I was very paranoid, 12 feet away from everyone around me. I was also not entering any buildings. And I want to show that all of us probably have good reason to be. 40 to 80% of people are asymptomatic or pre-symptomatic, and yet can be viral carriers and can transmit the virus to others around them. So the next slide gives you some statistics on this. If you could go to the next slide, thanks. Another statistic I want to share that really is horrifying is that there's 19 million people around the world who have so far been infected with COVID-19. My deep concern from this is that over 50% of those people have already neurological or neurovascular repercussions from it. I don't think the vast majority of deaths from COVID-19 will be this year or even next year. I believe they will be 5 years from now, 10 years from now, 20 years from now, because of the risk factors that the virus puts people at for neurological disorders downstream. So the next slide gives you some of the symptoms people are initially seeing when they have neurological symptoms. They include loss of smell and taste. They include sleep disturbances right away. They can include some milder things like headaches, and they can also include serious things like brain infection and inflammation. What I'm showing you on this slide is the neurovascular, physiological, and cellular changes that are ongoing with infection. So what is known is that there's a predominance of low oxygen levels in the brains of people who have been tested positive for COVID-19, and these are post-mortem samples. What that means is the virus is starving your brain, in a sense, of oxygen. Your brain is a voracious consumer of oxygen. It consumes 20% of your oxygen supply. The virus is not enabling your cells to be able to have its normal metabolism. It's also inducing what are called microthrombi, or clots that are forming in vessels, leading in perhaps to ischemia and microhemorrhaging. There's of course other changes. There's been reported stem cell changes and pericapillary changes and endothelial cell dysfunction. But the benefit of all of these is also they can be targets. So that's what this proposal is about, using what we know about the neurovasculature to actually target and allow us to be able to see changes in these particular metrics that we can monitor for pre-symptomatic and asymptomatic individuals and then track recovery. So the next slide gives you our two main goals, that is, use the same tools to detect COVID-19 non-invasively in pre-symptomatic and asymptomatic individuals, and then track neurovascular recovery. The next slide gives you a background of everyone on the team. There's a lot of people contributing. I'm very grateful for the NE mentorship and John, who's here. I don't have time to introduce everyone, but I can say with us, we have ophthalmologists, we have infectious disease experts, and of course, my lab member, Zach Maloney, who's on the call for questions. The next slide will give you a little bit of background about what we're doing. We're proposing to do something that is off the shelf right away to make a difference in the pandemic. So this is not a technology we're developing on our own. They're in-house methods, or they're available commercially. So wearables are one of them. Smell tests are another one. So non-invasive passive monitoring and continuing monitoring, and definitive results of smell tests, which are quantitative and allowed to be self-administered, would be at-home based tests that then allow us to have a no-go or go decision, so clear or quarantine decision for individuals while they're safe at home and not exposing other people. And then on-site, we're proposing OCT. It's a non-invasive, somewhat like ultrasound for tissues, but using light, ultra or near infrared light. And we were able to see at the cellular resolution with these images. And the goal is to be able to detect dynamics of changes of the neurovasculature with these retina scans. And then these are all screens that would then could be led to the follow-up of more definitive and specific tests. The next slide gives you where we are. So I'm going to just go through this slide really quickly, and I'm going to, in the next slide, show you in more detail what we have in place. So if you could go to the next slide, please. 7. So we are leveraging 150 or more now people who've been enrolled in a 3-year study that I've led in Houston and San Antonio and Los Angeles. They've been tracked most of them have been tracked 12 months or more, and a number of them have been infected with COVID-19. But that cyber infrastructure, the data analytics and AI pipeline is in place, and the coordination of the remote testing is in place. We have IRB approval for the study as well. With our team, we also have experts who have developed small and large animal models for COVID-19, and specifically they've developed rhesus monkeys models and baboon models, and also hamster and mouse models. The next slide gives you a little bit of background on some of the things that we're picking up already. We've shown proof of principle that we can pick up changes in REM duration over the course of stay-at-home orders or pandemic orders in individuals, and see distinct changes across the population level using sensor technology and AI methods developed in our lab. So the next slide gives you a background that we're not the only ones approaching this, and there's many, many different sensors out there that are available. Our mentor, John, really curated this list, and you can see a lot of the things on the left-hand side really illustrate that there's many devices, and we're agnostic to the device we use. The AI algorithms can be applied broadly. We're looking for changes across time in the neurovasculature specifically. So the next slide gives you a background on what we're doing at the cellular level. So the benefit of these retina scans is that you can see small changes in vessels like I'm showing here. It's a cotton wool spot, which indicates that this COVID-19 positive patient had ischemia in that location. And this seems to be very prominent in COVID-19 patients. There's another recent publication that's been out on it. But beyond this structural change, you also see quick dynamic changes. So prior coronaviruses have shown that there's changing the oxygen that's circulating within the capillaries of the retina. It also changes the leakiness of the blood-retina barrier, among other things. So the next slide, if you could turn to the next slide, shows you where we stand. for the smell test, I have in front of me, I was going to show it if I can next time, I have in front of me a Sensonics 8-item smell test. So this is a test developed by our collaborator, Dick Dottie at University of Pennsylvania, and he's shown definitively that in 95% of patients who are COVID-19 positive, that you can detect olfactory dysfunction. And this is something that happens very early on in the course of being infected, and it's more definitive, of course, than temperature checks. From the retina imaging, we've partnered with collaborators at UC San Diego, where they have also been able to obtain after a recovery from COVID-19, OCT retina images from a small sample of patients. And additionally, the methods for the AI-driven approaches have been shown utility in using retina scans in ischemia and diabetes. And we also have in-house neurovascular cellular analyses, image-based AI methods. The next slide, it shows you the timeline. So we're really trying to make a difference right away to impact the number of people who are infected by COVID-19, to minimize the number of people who are infected, and to ward off long-term neurological effects from this. So we want to have a 40-person trial right away this coming month, where we have COVID-19 positive patients and healthy volunteers. We also are developing the app for this. In October, we would like to have all of our animal models started. And then long-term, we want to be able to monitor people who've been infected by COVID-19 and scan for their brain health using the infrastructure we've developed. And eventually, we're also working on the computational imaging side to take what we're doing on the retina scans that are in these expensive on-site devices and bring it to the cell phone. With that, I'd like to close on the next slide on just saying that what we're doing here can save thousands of people per day from risk of infection. It also can enable early interventions to ward off what I believe will be many long-term repercussions from having a COVID-19 exposure and prevent strokes and other neurovascular disorders downstream. Beyond that, we also have parallel biomimetic models in my lab where we're developing retina models that can parallel the retina imaging and enable new therapeutic targets. So if you click one more, I'm just going to summarize what our ask is. We would like to have additional funding to expand our human subject studies, and then also to launch or to have a few animal model studies off the ground done in our BSL-4 collaborators lab at Texas Biomedical Institute. We would like to have dedicated imaging systems. We don't want to have COVID-19 infected patients using the same imaging systems for their analysis as those who are healthy. And then coordination. These are methods that can be done right away. So there's no reason why we wouldn't be able to do smell testing and use the AI algorithms to identify changes from wearable devices right now. It just requires the connections with institutes willing to do that for their employees. And that's what that ask is for. So thank you for the opportunity to share. I am happy to answer any questions. These are just backup slides. So it gives you a little idea what we're doing.
Thank you so much, Amina. For this round of Q&A, we have Paul, Darlene, Neil, John, and Sam. And our first question will go to Darlene.
Oh, great. So first off, that was a fabulous presentation and extremely informative. And as I went through it, it's a beautiful blend of both kind of real-time research, but also really trying to make a difference now. And I think with that, as I was listening, there was the smell test, there's the retinal piece. I initially thought the smell test might be the easier one if we're thinking consumers as the people that would actually, I'm going to say, be the customer. But it sounded like the retinal was where you were going. Is there precedent for cell phone retinal home analysis, so to speak? And is there any danger to people doing that 12 times a day to see if they think there's been a change? Or maybe you could say a little bit more about the implementation of that next step.
Thank you for these questions. These are actually very relevant ones we've been asking ourselves. So we want to do both. So it's not an either or. The small tests, you're right, they're available. So our collaborator, Dick Dottie at University of Pennsylvania, has a company called Sensonics where they have a commercial 8-item smell test. I have it in front of me, actually, if you can see it. And it can be used right off the shelf right now. And it's something that we want to be able to implement broadly because we believe it would detect earlier than anything like a temperature check could. And that's an easy thing to implement. So what we need there is just logistical connections with people willing to have this for their volunteers or for their employees. The retina scans have the benefit of that they're on site, so that you're not dependent on an individual saying that, oh well, the smell test told me that I might be at risk for COVID-19, but they still have the ability to move around the neighborhood or move around and go different places. So the retina test gives you an on-site based method right now, which there's commercially available devices for. And that's one of the benefits for it. What we would like to do is implement both. And then the long-term development process that we're doing is the computational imaging of the retina scans. And so your question is one that I've been really focused on the last couple of weeks. So AI integrated retina scans for the cell phone have been done before using fundus imaging. And there are some devices that are in development for what's called optical coherence tomography imaging. But the problem with the cell phone is that it would need another, right now, the current devices need another lens that would be expensive to add for individuals. So what we're trying to do is we're going to map in the same individuals, low resolution cell phone imaging, fundus scan, which is intermediate resolution, and this high resolution OCT, and then use computational imaging to map across these. If we're able to train them well enough, we would have enough of a sample size to be able to get the cell phone imaging in a way which would be able to be used at home. Any one of these are non-invasive and you can use them repeatedly. I've had an OCT scan myself. It's a 2-minute exam. It's very fast. It's like ultrasound. So there's no known risk factors for having this done repeatedly. So, and then the cell phone itself, if we do it that way, it would be really just using the light from your phone to be able to detect that changes in your retina. But these are really,
Excellent.
really interesting.
Thank you. Yeah, excellent.
Great. Thank you, Amina. John, next question is yours.
Yeah, Mina, I don't know if I really have a question, just encouragement for what you're doing. It's become more and more apparent recently that relatively healthy or mildly symptomatic patients can have significant vascular disease and concern about where that will lead. And it's great that you guys are focusing on this. I guess I do have a question that, because a lot of cardiologists talk to me about this. Is there, do you have collaborators that maybe could help you with blood tests or markers that could help follow people over time?
It's a good question. So already for the ongoing brain health study, we've been doing proteomic screens and genomic screens in collaboration with MD Anderson Cancer Center. And also UT Health Sciences and UTSA have in-house genomic screening. So we are collecting blood from volunteers right now, and I see no reason in not doing that for this project as well. It's a good question because we want to have therapeutic targets. Our experimental work also uses the blood to develop these brain models in a dish. So for us, collecting blood allows another opportunity to look at functional changes in the cells from the volunteers that we're collecting the blood from.
Great. Thank you.
Thank you. Next question goes to Sam. Sam, you're muted.
My question was, have you looked at food test?
The taste.
Like smell,
food.
There is also food test.
I'm sorry, food like taste.
Eating food.
Yes. Our team has not done that yet. It is something to think about as well. I think that might be more difficult to implement.
But it's a simple test. It's a very simple test. You know, I have talked to several friends and doctors, and they believe that along with smell, food test also changes.
Yes, that would make sense from logical effects of COVID-19.
That may be simple to do.
I'm sorry, and it's simple to do?
It may be.
I would like to hear more. It would be very interesting. I definitely have to read more about it. And if you have any connections there, it's something, and we can implement it quickly.
Okay.
We have volunteers.
Okay.
Great, interesting idea. Our last question goes to Neil.
I'm intrigued by your presentation. I think there's some really exciting things going on here. And if one is able to detect COVID-19 early by some of these methods, are there therapies that, the most dangerous thing about COVID-19, as I see it, is the long-term damage that it can cause to not only the brain, but to other organs. So early detection, if you can apply rectifying therapies earlier than might otherwise happen, is this likely to offset those long-term effects?
That's absolutely where we're headed. So along those lines, the brain health study that I conduct also is after preventative medicine. So we're looking at ways you can ward off strokes, or you could ward off age-related dementia. I believe viruses have a long history in how they might increase the risk for long-term health ramifications in the brain. And so there has been studies, and our work is looking at ways to prevent that, and compounds that can be used, or even behavioral changes. But without detection, without knowing, and without tracking, you don't know the people at risk. So that's exactly why we're doing this project.
I think that's marvelous. Thank you.
Thank you very much. Thank you, reviewers, and thank you, team. I'd like to bring up our next team.
Thank you, Alice.
Thank you. Our next team is combating misinformation on COVID-19.
Hi, everyone. My name is Locke, and today I'll be presenting Verify, our solution to combating misinformation on COVID-19. This is joint work with Krishma and Yan, based on our research at the USC Viterbi School of Engineering. Next slide. In addition to COVID-19 pandemic, the steady rise of misinformation known as the infodemic is also very dangerous. The impact of this can result in the loss of lives, as seen in the second or maybe even third wave, as people ignore safety policies and downgrade risks due to false information. Next. As you can see, not only in textual formats, misinformation is also being widely spread via video and social media platforms. Next. Society as a whole is facing challenges, which are perpetuated by the wide-reaching capabilities of social media platforms. Not only that, these platforms are also facing major challenges in detecting and mitigating misinformation in their data and in their networks. This can lead to users questioning the integrity of their platforms, the quality of their information, and subsequently leading to advertising partners withdrawing from their services, or worse. Next. As with recent advances, our solution leverages advanced ML and AI techniques, which have been developed in Yan's lab for the better part of a decade to combat misinformation. With the expertise in our lab, we have developed and commercialized novel frameworks to detect misinformation early within the networks, using user responses using Neural User Response Generator. We also use the capture score and educator module to help capture information from their source. Moreover, to attack the problem at the core, we identify and uncover a malicious group campaign. Sorry, I forgot to say next. Sorry. We want to identify and attack the source of the problem. We identify and uncover malicious group campaigns between coordinated accounts. We propose to mitigate misinformation by inferring and limiting the diffusion spread within the networks. And lastly, we also develop and interpret models to take on the challenges of detecting fake and generated videos. Next. Our approach to this deeper reaching problem is not only to detect misinformation, but also to identify the originating source and to mitigate the spread of these malicious contents. Moreover, we continuously monitor the impact of misinformation and the public sentiment towards news and policy. It helps us educate the public about facts relating to COVID-19. Next. We develop a dashboard that monitors malicious tweets on Twitter via the open source API, along with categorizing tweets that are incorrect, unreliable, clickbait, or conspiracy. Next. In addition, we monitor the spread of these tweets across countries to pinpoint the sources and geoinformation of the first time a misinformation was tweeted. Next slide, please. The dashboard processed tweets on Twitter from March 1st to June 30th, and we were able to detect at least 150,000 problematic tweets. We also monitor country-level sentiment towards various aspects of COVID-19 for 121 countries across the globe. Next. The value that our system brings to the table extends beyond COVID-19. It deals with a more general problem of misinformation. We know that misleading articles and fake news are textual domains that can be used to smear political candidates. However, on top of that, the recent development of deepfakes in videos and images domain also poses a great threat. Our solution can help mitigate this by detecting and limiting the source of information while also learning genuine signature of content that needs protection. Next. Our team possesses unique expertise in various information domains, along with years of industry experience. I myself have been working on deepfake generation and detection both at MIT and since the start of my PhD. Karishma is an expert in social network analysis and has been working with information cascade and propagation dynamics for the better part of the last 5 years. And Yan is a renowned professor and expert in machine learning and AI techniques for social goods. Next. The next step for us is to make available the COVID-19 analysis online by August 15. We will finalize and release a video module to showcase the capabilities of mitigating misleading videos by October 15 for the upcoming election. And by November 15, we would like to demo for customers with specific use case, which will require additional training and testing. And by December 15, we wanted to make available our system online. Overall, we believe that to combat misinformation, large-scale AI techniques like ours is the right way to take the challenges head-on. Next. Our dashboard is available at the URL within the slide. Thank you for your time, and we would be happy to take questions.
Thank you so much, team. Our reviewers for this Q&A session are Larry Seifert, Kurt Peterson, Sam Petroda, Monty Alger, and Darlene Solomon. And the first question will go to Sam.
I'd like to talk to you about application of this in politics, election. I think it's a very important tool if it works well. I don't know how well it works. So I'd like to connect with you later on because I think the application is huge.
Yeah, thank you so much for providing this opportunity. We agree with you. This is a really timely, very urgent application of AI to address some of the most important challenges that we're facing. The upcoming presidential election, we're really worried because compared with 4 years ago, the bad actors have been utilized a lot of more sophisticated strategies in order to confuse people, discourse the misinformation, and hurt the whole society. We'll be left to getting connected and work with you. Thank you.
It's not just misinformation. I think it's lies. And if you can tabulate all this and present it to people of all kinds, all over the world, it's not just a US problem.
I agree, right. Yeah.
Thanks, Sam, for making the connection. Our next question will go to Kurt Peterson.
Yeah, thank you very much for a great presentation and a great idea. I agree with Sam that this could be enormously useful data. My question is around how are you going to get, one of the problems with sites like this is getting yourself advertised and getting yourself out in the mind's eye of all the population of the U.S., for instance. So how are you going to try to get yourself known among the community for this misinformation on COVID-19?
Yeah, thanks for the question. This is a really challenge we're actually facing. We have made a few attempts. So one is that actually as a member of the New Voices for Academies of Science, Engineering, and Medicine, we are actually a group of 20 young research scientists in the whole field. So this project is actually in collaboration with that organization. In addition to building this particular dashboard for the misinformation, we also collaborate with the organization to build a chatbot in order to advocate what is actually facts about COVID-19. So we think that working with organizations or local governments in order to actually monitor the misinformation and also broadcast what is really the truth about COVID-19 is really important. And the second attempt we have actually made is working collectively with the reporters and the media. We think that they have a really good weapon in terms of how to combat this really confusing information online. So we have been talking with reporters from Fortune magazine, where they actually featured our work in their newsletter. In addition, we also talk with people from NPR and a few other media organizations. So we think that this could be another way. And obviously, we should also talk actively with the social media platforms. We are trying to get in touch with a few internal people within our organization, but this is still a relatively weak link. Obviously, last but not the least, we also publish papers ourselves in order to get reach to the majority of the general public. But we have to acknowledge this is still a weak link on our end, and we hope that anyone is interested in helping us to get the words out, and in order to help the society, please get in touch with us so that we can really help together. Yeah.
Thank you.
Great, thank you. Our next question goes to Monty.
Yeah, hi, thank you very much. It's a very interesting presentation. A couple thoughts and then some questions, and I'd like to also follow up with you. First of all, this question of brand and how are you going to become the place to be visible? If you could take some of your results and convert them into lost money spent, number of unnecessary deaths, something that takes the result of misinformation and converts it into something that's accessible to the public, so we can get a handle on this. You also have, you can do this for COVID, but you obviously have a platform for doing this for any misinformation anywhere. And that's sort of a general problem. And I would imagine you've been thinking about how you might apply this more broadly, not just to COVID. One thought I'd suggest is possibly building some kind of online class for use with universities or others to teach about misinformation, and then share your tools and the technology you're developing so others can engage with this, because this problem is only going to continue to accelerate and get worse.
Yeah, thank you for the wonderful suggestions. We totally agree that getting a specific set of tools to help people understand what is misinformation, the impacts, and also how to combat as a general society, I think is really important. Yeah, education is a key component for everyone to actually get educated about the impacts. That's a really good suggestion. We probably will think very thoroughly about this direction and then maybe pursue in the longer term. Thank you.
Thank you.
Fantastic. Thank you, team, and thank you, reviewers. I'd like to introduce our fifth team now. This is Remote Wearable Monitoring System for COVID-19 patients.
Hi, I'm Christine. I'm with Sense4Me. And today I'm going to be introducing a wearable patch that will help doctors remotely track COVID disease progression and specifically the immune response that patients are exhibiting. Next slide. So COVID got personal for us. This is our friend, Katerina. The team knew her. She caught COVID and she had a pretty mild case in the beginning for about a week and a half. And then on the 9th day, she developed very severe disease all of a sudden. And then she was in the hospital for 2 weeks and she ended up passing away. So, you know, going through this experience with her, we were pretty shocked that she was, you know, feeling fine, and then all of a sudden she got so sick. So that was, she was our motivation really for kind of finding out why that was the case. Next slide. So digging deeper, talking to clinicians and researchers, we found that this period of time when she was experiencing, or anyone that has a severe disease, she was experiencing these mild symptoms, but actually there was a so-called storm brewing inside of her. So inflammatory markers were elevated, and we found out that ultimately what people are passing away from with COVID is that they have an overactive immune system, which is caused by these high inflammation levels. So we kind of identified this period of time when patients are at home and they have mild symptoms. There's no way of really tracking what's going on, that this is a very critical stage in the disease. Next slide. And currently, these 19% of patients that are going to develop severe disease, there's no way of knowing who that's going to be based on the current system in place. So that system is you get a diagnosis, you get your temperature measured, when you go back to the doctor, you get oxygenation levels measured, they ask you how you're feeling, but that's pretty much it. And vitals don't measure inflammation, so they're not giving the full picture of what's going on. Next slide. So our solution is this patch that will measure autonomic response as well as stress and inflammation markers. And this data will be collected and then transmitted to an app and a platform that doctors will have access to and patients, but the doctors will be getting insights based on this data. Next slide. And what we aim to do to help them do is to improve treatment timing, because that window that we talked about, where currently patients are home, there's no way of really finding out what's going on inside. The doctors will be able to administer a drug or call them into the hospital earlier if they see something that's of concern. And ultimately, this will lead to reduction of hospitalizations and reduction of mortality. Next slide. So this is our team. Lily is our chief. We have multiple components to our solution. So each of us covers a different aspect. Lily's our chief technology officer. Nasir is our medical advisor who's affiliated with Columbia University. Darnell is spearheading the data science and software development, while Tyler is working on the device aspect, and I am the lead on the business development side. Next slide. Currently, we are finalizing our prototype, and going forward, what we will need is help on our business model development. So who exactly is going to be using this product, how do we get it out there into the market quickly and efficiently, and regulatory advisory, so what it's going to take to get this approved and out there. And then finally, a manufacturing partner to help us scale up and provide everybody that needs this product with this. Next slide. Thank you for listening to our presentation. This is our contact information, and we are more than happy now to answer any questions you have.
Great, thank you. Our reviewers for this session are Paul Citron, Monty Algier, Dennis Disher, Larry Seifert, and Kurt Peterson. And our first question will go to Dennis Disher.
Yeah, hi, thanks a lot. I think the motivation for this is very, very strong, as you say, to try to detect early. I was wondering if you could give us more details on this patch. In particular, let me just ask, are you trying to access blood as well, or is it just superficially on the skin?
Let me take that question. We have, and thank you very much for the question, because we did not provide enough here. So we have currently developed a prototype to address the autonomic system response, more specifically measuring skin conductance. The prototype is collecting, we are able to collect data now, and the data is sent to a secure database, and we are building that now. We have proof of concept of the stress biomarkers and ultra-flexible rate. We are collaborating with EPFL in Switzerland. And we have underway the proof of concept of the inflammation biomarkers from the same platform. And it's really just on the skin and receiving information from the sweat. Now, luckily, these markers have about the same, and actually in some cases, a little higher concentration in the sweat than in the bloodstream. So in terms of innovations that we have accomplished, we have a scalable prototype for the autonomic response that involves additive manufacturing, and more specifically 3D printing of metal interconnects. We are amplifying the signals of the biomarkers of stress and inflammation with nanostructured coatings on the electrodes. And we are developing algorithms to predict disease severity. I hope I answered your question.
Yes, thank you very much.
Our next question will come from Kurt.
This is a very exciting development. Actually, this has been one of the problems, as of course you know, with this virus is it's just unpredictable in terms of whether people are just going to react mildly or dramatically. So this is a great application. I love it. I have two questions. Have you already identified, and it seems like you have, the biomarkers that you're using? And are these kind of just generalized markers, or are they specific to COVID? I guess in a sense it probably doesn't matter, but I'm just curious about that. And then, secondly, what about correlating with SpO2? I would think that would be a very good measurement for inflammation. So those are my two questions.
Yes, great. So let me try to answer. Let me take that question as well. Yes, the more specific, there have been many studies primarily from China and more specifically from Wuhan that have been published, and some other studies from Singapore that have tracked patients while in the hospital, but for extended period of time, from day 1 of their symptoms to day 40 or 50, when the symptoms and the disease resolved. And what they have found is that in the case of severe disease, there are certain levels of cytokines, which are pro-inflammatory cytokines. They are proteins that are implicated in the immune system, and they are really the messengers they enlist, they encourage the immune system's cells to attack the virus. And these pro-inflammatory cytokines are primarily the ones that are markers for the disease condition. Now on to your second question. And by the way, these pro-inflammatory cytokines are implicated in other disease conditions, more specifically in autoimmune conditions. Certain autoimmune conditions, for example, lupus, rheumatoid arthritis, and some other autoimmune conditions. That's why some of the medications actually from these diseases to COVID. So on to your next question, definitely having to integrate the oxygen level will be a great thing to have. But as a starter, we have the skin conductance, which is indicative of the breathing. It can actually measure breathing, especially if the breathing gets heavy. And we have that done. We are looking into that.
Okay,
fantastic. Thank you. Thank you so much. Thank you, team, and thank you, reviewers, for the insightful questions. This concludes the pitch showcase portion of our program today. Please join me in congratulating each of these teams on the impressive progress that they've made in such a short amount of time. Congratulations, teams. I'd now like to invite the chair of the Expert Review Committee, Paul Citron, to share some remarks on the presentations today. Paul, over to you.
Thank you, Alice. You did a great job moderating once again, so thanks again. Engineers are problem solvers. The National Academy of Engineering COVID-19 call for engineering action is intended to harness the creativity of engineers, whether they are still students in academia, industry, or retired. It's a multi-generational effort. Engineers have in their DNA to make things better. They drive useful progress. They apply their ingenuity to essentially all aspects of life. Sometimes they make breakthrough innovations. More often they make useful and valuable advancements that build on earlier innovations. It's what engineers do. The sudden emergence of COVID-19 was, and still is obviously, a game changer. It poses a serious threat, sometimes deadly, to those who contract the virus. COVID-19 has overwhelmed hospitals around the world, exhausting caregivers, and unfortunately infecting many of them. Its impact cuts across many other aspects of society: social interactions, travel, the economy, jobs, education, critical supply chain integrity, disease resilience, disaster resilience, and patient access to non-COVID healthcare. And the list goes on and on. COVID-19 has spurred this initiative to develop more effective approaches for detection, treatment, mitigation, and ultimately prevention of the disease. Please note that today's session is not a culminating event. Rather, it is the second in a series, and our idea incubator is ongoing. I'd like to, if I may, take a few minutes to provide my summary observations relating to today's presentations. Team 1, self-disinfecting mask. Locally enhanced electric field treatment, LEEFT, is an emerging breakthrough technology for disinfection and microbial inactivation. Applied using low-power nanowires, it may provide a practicable way to safely disinfect face masks, thereby extending their useful life. Team number 2, wearable patch for remote monitoring of COVID-19 patients. COVID-19 has led to enormous overcrowding challenges for hospitals and their staffs. Strategies that permit patients to safely stay home could ease overcrowding. The monitoring system proposed by this group may be a way to accomplish this. For it to be useful and adopted, it would need to integrate and translate the raw data it collects into actionable information regarding a remote individual's physiological trends and status. Raw data transmission may not be sufficiently useful. Team number 3, early COVID-19 detection and neurovascular recovery in pre-symptomatic and asymptomatic individuals. COVID-19 seems to affect the nervous system in some survivors, and this may have implications for cognitive decline, particularly in older individuals. The creation of a crowdsourced COVID-19 resource and recovery website focusing on cognitive health would be a welcome tool for improving long-term outcomes. Team number 4, combating misinformation on COVID-19. Misinformation regarding COVID-19 is rampant. A challenge for this information dashboard will be to gain the public's trust. There are algorithm-based screens that identify and label what is misinformation versus, let's say, well-intentioned opinion is viewed as being critical to acceptance. An interesting aspect of this dashboard is its ability to spot trends of emerging hot topic issues and measure their intensity. This marker could provide a way to more precisely and forcefully respond to serious misinformation. Team 5, remote wearable monitoring system for COVID-19. In comparing the presentation to the abstract that I had, I see that the concept has matured and evolved. And what I would really advise the group to do is to focus on the sensors that are readily available rather than having sensors that are on the critical path that have yet to be proven. For those in the audience who have inputs regarding any of the pitches or would like to help this initiative in any way, please contact the NAE at engcovid@nae.edu. If you would like to enter the call to action incubator with ideas, please go to nae.edu/COVID-19. I'd like to thank today's teams for responding to the challenge and proposing a piece of the puzzle to mitigate the effects of COVID-19. My thanks to the Expert Review Committee for their insights, Alice for moderating, and also to the NAE staff, including Brandon Green and others behind the scenes who have helped put this event together. We hope to host you again in the future at future rounds of the pitch. Thank you very much.